So as I was listening to the introductory talks from Heather and Kathy, there is quite a bit of overlap between our disorders and probably a lot of shared learnings as well. So Rett syndrome affects about 1 in 10,000 females. It's almost exclusively diagnosed in girls. There's a genetic reason for that I won't go into, but in rare cases, it is also found in boys. The gene that's affected is called MECP2. It's on the X chromosome. Girls have 2 X's. One of them is silent. And so a girl with Rett syndrome has about 50% of her cells that have the mutated protein, and the other 50% that have normal protein. That gives you full-blown Rett syndrome. In the case of the boys, they don't have that backup protein. So they have no MECP2 or no functioning MECP2. And so they're typically much more severe. There's no at-risk population. You find the same incidence regardless of ethnicity or race or any kind of socioeconomic factors. It is mutations in a single gene, which is good because it means we have a clear target for intervention. There's been about 900 mutations that have been identified, although there's about a dozen or so that encompass about 50% of Rett cases. It affects nearly every symptom in the body, and I'll use my daughter as an example. She's 27. She can't speak, she can't walk. She has intractable seizures, fed through a feeding tube, cannot use her hands at all. In fact, loss of purposeful hand use replaced by a stereotypic movement is the hallmark symptom of Rett. She also has problems with sleep, breathing irregularities, breath holding and hyperventilation, and anxiety, scoliosis. In nearly 100% of the patients, about a third undergo scoliosis surgery. So it's a very severe disorder. The kids grow up to be adults and can live many decades, but quality of life is, pretty hampered. It's not neurodegenerative, which is good news. That means that the brain cells are not dying. And we do not have any effective treatments. Our kids, like your kids, are on sleep medications, antidepressants, anxiety meds, seizure medications, GI. Oh, and I didn't mention GI. They have a lot of GI issues as well. The gene was discovered by Kathy's husband, Adrian Bird, in the early 90s. And then in 1999, it was discovered by Huda Zogby at Baylor in Houston that mutations in MECP2 were causing Rett syndrome. That was around the time that I became involved. My daughter was diagnosed in 1998. So a year later, the gene mutations were found. And I started a nonprofit called the Rett Syndrome Research Foundation. And that was a really good time to get involved. Because the gene had been discovered and we were able to pull into the research people like Adrian, Rudolf Janisch, and others. We already knew a fair amount about the gene. We started off at RSRF. I was lucky enough that I was able to start the foundation with a handful of other parents. At the time, there already was a nonprofit, but they were family support focused and were not that interested in research, and suggested that I go start my own organization, which I did. And in the beginning, we didn't have a whole lot of money, and we were providing small grants to academic researchers in lots of different fields, because we really needed to kind of fertilize the field. And so we were funding biochemistry work and genetics and some clinical genetics and, lots of different areas to try to understand what the protein was doing, where was it found. We started to make animal models, a variety of different animal models to serve different experimental purposes. We were also growing our network of advisors. We were doing a yearly request for proposal. That's when we started out, was maybe we were maybe getting 10, 15 proposals. And with every passing year, that number grew. At the end, we were getting over 100 proposals for every RFP. Of course, that meant we needed a number of reviewers, key opinion leaders. So trying to grow that network. We were hosting an annual scientific meeting where we were bringing together the various labs. Again, it was all academic at the time, together for a couple of days to share data and to brainstorm and to really advise the Rett Syndrome Research Foundation on where we should be investing our funds and what could we do to stimulate more research. In 2007 came a landmark paper, again from Adrian, Kathy's husband, that showed that if you can restore levels of this protein, the symptoms in a mouse model dramatically go away. Everything that was measurable went back either to normal levels or almost normal levels. That was pretty unexpected. I don't know if I would say that it was unexpected for me. I was hoping, of course, that would be the outcome. Maybe it was just my need to have that be the outcome as a parent of a child that, was severely disabled. But I think it really shocked the rest of the scientific world, clinical community, and of course the parent community as well. And I think it's become kind of the reversal, the experiment to do, and has been repeated now for lots of different disorders. Not always with the same results, of course. I think Rett stands out as a disorder that's really remarkably reversible, at least in mouse models. We'll have to wait and see how that translates to people. I ended up leaving the Rett Syndrome Research Foundation for a variety of reasons and started the Rett Syndrome Research Trust in 2008. And the goal of the Rett Syndrome Research Trust was really to focus on genetic medicines. We have three main reasons for optimism. One, it's a single gene disorder, like your disorder is. The second is that it's not degenerative, right? The brain cells are all there. They're all in the right place. They're not communicating with each other properly. They're not functioning properly, but they're all there and they're not dying. And then we have this prospect of reversibility, which gave the community a huge boost. And for me, it really changed everything. It made me really want to go after genetic medicines. Now, at the time, 2008, the technologies still weren't there. It was difficult to deliver to the brain. It still is difficult, but at least we have some technologies that allow us to deliver genetic medicine cargoes to the brain. And so it just really made a lot of sense to go after genetic medicines as our focus. The field had matured. We knew a lot more about MECP2 and about Rett syndrome clinically as well. As an organization, we moved away from RFPs, requests for proposals, and moved towards, deciding which areas we wanted to be focused on. So, for example, we decided very early on that gene therapy was going to be a focus for us. Genome editing and RNA editing really hadn't been discovered yet. So there was really one way to boost levels of this protein, and that was through gene therapy. So we decided that was really going to be a focus for us. We had other areas of focus, but the way we still operate is not to do an RFP, but to decide the areas that we want to focus on, figure out who's doing cutting edge work in each of these modalities, and then cultivate a relationship with these scientists and encourage them to submit proposals to us for funding. That's a big part of what I do day to day is really outreach, whether it's to academic labs or to industry, to really try to stay on top of who's doing what in the field and making contact and cultivating relationships with anyone that's doing work that's relevant for Rett syndrome. We started funding gene therapy work in 2010, and we funded a number of labs. And in 2013, there was another key paper from Gail Mandel, Brian Kaspar, and Adrian Bird, showing that in female mice who are lacking MECP2, that we could restore or reverse symptoms using gene therapy. So that was the first solid clue that gene therapy was something that could be relevant for patients. So at that point, I kind of took stock of the gene therapy landscape for Rett syndrome, who was doing what, and decided that it would be helpful to have a gene therapy consortium, because the situation that we found ourselves in, and I think this is common, is that you've got gene therapists who know a lot about how to deliver genes to the body. That is their area of focus, but they don't know anything about our particular disease. And then you've got people that know a lot about the disease, in our case, people like Adrian, for you, Kathy, and others, but they're not gene therapy experts. And so the thought here was, can we bring people together in a consortium with synergistic but different skill sets and get them to collaborate, provide funding, provide the infrastructure, provide opportunities to meet in person, provide opportunities for lab members to go to each other's labs and spend time and learn new experiments and techniques and share ideas. And we did that. We started a gene therapy consortium with 2 MECP2 experts and 2 gene therapists, and funded them from 2014 to 2017. And that was a very a great investment, because out of that gene therapy consortium came both of the gene therapy companies programs that are now in companies. We transitioned them from academia to companies, Taysha Gene Therapies and Neurogene, and also an RNA editing program that started in Gail Mendel's lab and now is in a company as well. We're still focused on gene therapy, but we've also expanded now to other technologies, which I'll touch on in the next slide. As the science began to mature, we also started to have more engagement with biopharma. Typically, that engagement starts with me doing the outreach. Occasionally, we get contacted by companies as well, but it really takes quite a proactive approach of reaching out to companies that are in the gene therapy space or in the genome editing space, base editing, prime editing, these various ways to not supplement the gene by adding extra copies of the gene, but actually by correcting the mutations. The other thing that we did as the science matured and as we started raising more money was being able to hire staff that had industry experience. So when I started RSRT, it was pretty much me, but fortunately, we were able to hire people with industry experience, genetic medicine experience, clinical operations experience, and translational medicine. So we now have a team of 5 focused on the research. So these are the genetic medicine modalities that we're working on. Gene therapy, of course, is the furthest along. It's the technology that's been around the longest. There are 2 gene therapy trials, as I mentioned, 2 companies, actually multiple gene therapy trials, but sponsored by 2 companies. Still too early to tell, how well it's working. We're hoping that by the end of the year, both companies will be in a position to share some data. We're also working on base editing. So that's where we're able to address certain mutations that are caused by a single, point mutation in a single nucleotide. Prime editing. So base editing was discovered by David Liu at the Broad Institute. He also discovered prime editing. I describe prime editing kind of as base editing on steroids. You're able to fix multiple mutations that are close to each other. Right now, that means really close to each other within, a couple of amino acids. But in time, this should be, as the technology matures and people get better at doing it, it should be able to address, entire, large sections of DNA, maybe entire exons. Because MECP2 is on the X chromosome, another approach of ours is to try to reactivate the silent MECP2 that's on the inactive X. And then we have RNA approaches as well, RNA editing and RNA transplicing. In transplicing, we're trying to splice out the 2 exons that have 97% of all of the mutations in MECP2 and splice in healthy exons. With the exception of MECP2 reactivation, these are all technologies, modalities that are applicable to your disorder as well. Then on the clinical research front, patient data and being able to assess symptoms in an objective fashion is also really important. We recently launched a Rett syndrome global registry. And so this is a registry where it's parent reported. So parents register, they register themselves, they consent on behalf of their child. And then they're asked a series of questions that we developed about symptoms and history, medical history, surgeries that they've had, also what doctors they see, therapies that they get, medications that they're on, what medications they think are helpful. So, a series of questions, and then they're prompted quarterly to go in and update. And then we have this digital natural history study, which doesn't cost anything, and it's something that I would encourage your group to look at. It's a company called Ciitizen, spelled CIITIZEN. As I mentioned before, they had been bought by Invitae, but then spun out and they're independent again. So it's a technology company, and the way it works is parents register on behalf of their child. They have to go through a registration process that proves they are who they are, and then they give consent for the company to solicit medical records from physicians. And you put in the doctors that your child sees, or the clinics that they attend, hospitals, whatever it may be. The more comprehensive the information you provide, the better it is. And then Ciitizen requests the medical records on your behalf. And then they have a machine learning program that allows them to extract information from, structured information from unstructured medical records that might be in PDF form, for example. And then the data gets shared back with the nonprofit anonymized in a spreadsheet form. And the thought process here is that if you're doing a traditional natural history study and you're doing a 10-year natural history study or a 15-year natural history study, that takes 10 years or it takes 15 years. And you're only getting a subset of people. You're getting the subset of people that are willing to participate, that can travel to a clinic, whose children are not too sick, or it excludes families that can't afford to take the day off to go to a clinic, or can't travel to a clinic. So there's pros and cons of each approach. We have a traditional natural history study in Rett. This is meant to complement that and to add additional information, not to replace it. But this allows you in a very short period of time, as long as you can recruit patients, to get deep knowledge from the medical records. Because imagine how much information exists already in our kids' medical records. So Ciitizen is something that someone could contact, and if anyone is interested, I can provide, I can give you the contact information of the person to reach out to. Our global registry we built with a company called Across Healthcare, and their registry program is called Matrix. This is a company that was started by someone who is very passionate about rare disease. He himself has a rare disease. And they kind of charge you on a sliding scale. So there are many patient advocacy groups that are for rare diseases that don't have a lot of money, who are able to build a registry almost for nothing, or very little money. And then organizations like ours that have a bit more money are able to subsidize because we're paying at at a higher level, and happy to do that, actually. So again, if someone is interested, I'm happy to make an introduction to Across Healthcare. Biosensors is an effort that we're undertaking to try to find objective ways to measure symptoms directly from the patient. What has happened in Rett syndrome is that the outcome measures that have been used in trials have historically been very subjective questionnaires that either parents have to respond to or physicians. And because they're subjective, it makes them very placebo vulnerable. And we're looking to replace these questionnaires with objective outcome measures that are actually collecting physiologic data directly from patients. Things like sleep, seizures, breathing, movement. And so we're testing a number of devices, and we're in the process now. We're just launching a study that's going to be testing a device that we're very excited about, and that so far we've done a pilot study with, is something called the Emerald. It's a box that sits in a room, kind of like your Wi-Fi box, and collects data through radio waves. Our pilot study was focused on sleep and on breathing. And of course, they have to build algorithms, but it was really, really encouraging. And so now we're expanding that with the number of, expanding the number of patients. We're also going to be characterizing movement, and we will be comparing sleep and breathing using the Emerald while people are undergoing overnight sleep studies, which is kind of the gold standard. And so we'll be able to answer the question, is what we think we're collecting with the Emerald actually what we are collecting? So we're excited about that study, and that's getting underway now at 3 clinics in the US. We also have a biomarker project, and the hope here is to be able to find something in blood that tracks with disease severity, and would be able to give you an early signal of target engagement. So for example, in gene therapy, you might imagine that seeing improvements in gene therapy is going to take some time, because it requires, a patient to learn skills. And a biomarker would be able to perhaps tell you early on whether you're on the right track. There's no guarantee that we're going to find one. We have some candidate biomarkers that look really interesting, and so we're chasing those. I think if we had a biomarker, it would make clinical trials easier. I think we'll see many more companies getting involved, but we'll see. There's no guarantee that we're going to find it. And I think biomarkers for neurological diseases overall has been a challenging field. And then finally, we have a biorepository. So these are patient cell lines, fibroblasts, and then a subset. We have 30 patient cell line fibroblasts, but then we also have 5 and soon to be 6, actually 7, the patient cell lines that we've made iPSCs, induced pluripotent stem cells. And we've had over 100 requests from companies and academic labs. The reason we made a biorepository ourselves is because although there were plenty of patient lines that had been developed in academic labs, we found that companies couldn't get a hold of them. So even if the academics themselves wanted to share them, it was difficult. Their institution wouldn't allow sharing in most cases with companies. And so we collected our own patient cell lines and funded the, making these various lines and iPSCs, and we're maintaining the repository at Coriell and handling the MTAs and things that are required. But it's been very, very helpful. So those are the clinical research efforts that are underway. I feel really strongly that organizations like ours are really important to get drug development and genetic medicine development moving in rare diseases. This kind of work just doesn't happen on its own. It's one thing if it's Alzheimer's or it's some other major disorder where there's a lot of incentive for companies to get involved. But for rare diseases, you really need somebody that's like driving these efforts forward, providing funding to labs and to getting companies engaged, and that laser focus on a cure. And really what we're doing is de-risking programs. So no company, Rett syndrome, and I suspect your disorder too, it's just not low-hanging fruits for companies. And there's tons of rare diseases. And, for companies to get involved, they ask a number of questions. How much is known about the protein? Are there animal models? Are there patient cell lines? What would we use for outcome measures? What could we recruit for a trial? Are there clinic sites? Is there a biomarker? And the more of those things that you can check off and say, yes, we have that. Yes, we have that. Yes, we've got patient registry. Yes, we have clinical data. Yes, there is a natural history study. The more likely it will be that companies, could be convinced to pick up a program. And so I think what we do is we de-risk these programs. We get them started in academic labs. We build the resources that are required, and then we help to try to make that transition from an academic lab to a company where drugs are typically developed. When I say drugs, I mean genetic medicines as well. We are technology agnostic at the end of the day, whether it's gene therapy or editing or something else that works. We want to see all of these efforts move in parallel forward as fast as possible. We think the more shots on goal we have, the better the likelihood of success. And we really act as matchmakers, between academics and industry. Cargoes and delivery, what I mean here is our genetic medicine cargoes, whether it's gene therapy or the machinery to do editing or RNA. And then for now, in terms of delivery, the gold standard is AAV9. That's what the 2 gene therapy companies are using, and that's what lots of academics and companies are using. In time, we hope that we'll be able to transition to a non-viral delivery method, because there are a number of advantages, immune responses being one of them. For now, you can only give AAV9 one time. Non-viral, and by non-viral, I mean things like LNPs, lipid nanoparticles, which is how the COVID vaccine is delivered, or exosomes, or some other non-viral delivery. They're cheaper to make, they're easier to make. There's no cargo space limitations as there is with AAV, and no or less immune issues. So there's a lot of advantages. Lots of people are trying to develop delivery technologies that get into the brain. We're talking to a number of companies and academic labs. We just started a delivery consortium to share ideas and to advise us on where we should be investing some funds. So I'm optimistic, but we're not there yet on the delivery. For now, we're depending on AAV9, and we'll have to see how well that works for our disease. And in terms of the clinical research efforts that I just described, the registry, the natural history study, I think those are efforts that are important to be advanced by a nonprofit who's going to make sure that everyone has access to them. If it's a particular biopharmaceutical company that develops, it ends up being proprietary and not shared. And so I think it's important for all of us to work on those and to make those resources available to anyone. So of course, that all sounds great, but it takes money, right, to fund all of this work. And I wish there was a magic bullet, but there isn't. All of our fundraising is really, grassroots, and the money that we have raised, every single penny of it can be traced back to families. That doesn't mean that all of our money comes from affected families themselves. Some of it does. The vast majority comes from their network, family, friends, colleagues, relatives, neighbors. And so we really depend on families to fundraise for us. And it's not easy, right? Because families already have a lot of challenges that they're dealing with. But unfortunately, that's the case. Anyway, I hope that was helpful, and I'm happy to answer questions.